Alright, so in this video we're going to talk about meiosis as compared to mitosis. There's some big differences between meiosis, the main of which is that meiosis is all about creating genetic variation. Mitosis created identical cells, but meiosis is going to form variable sex cells, right? So there's three things that contribute to this variation. We have crossing over, random alignment of the chromosomes, and random fertilization. And these three things should show you that you being you occurs at an extraordinarily low probability, which the probability of nature is going to be represented by this dice right here, or a die, it's singular. Alright, so from the last video, we went through mitosis. We went through the whole shebang, and we're now going to begin meiosis following DNA replication. So we're going to have already gone through S phase, we're at G2, but instead of mitosis, we're going to go through meiosis to create sperm and egg, or one or the other. We'll see what we got. Okay, so bringing back our sock models, we have a Y chromosome and an X chromosome. So we're going to be creating sperm because this parent is a male. Alright, so note that the blue sex chromosomes are Y chromosomes, the pink sex chromosomes are X chromosomes. However, the autosomes, so this pink autosome is maternally inherited. So if we're thinking that we're making sperm from a soon-to-be father, this comes from the grandmother, right? So there is a pink autosome. Here is a blue autosome, a homologous chromosome, because they're the same, but one's maternal, one's paternal. And then same with the larger autosome. We have this one from grandma, and we have this one from grandpa. Okay. So in total, we have 6 chromosomes and we have 3 homologous pairs, right? We consider the X and the Y to be homologous pairs, even though they're not the same. They're the sex chromosomes. Alright, so yes, blue comes from the sperm that created this soon-to-be father, and pink comes from the egg that created this soon-to-be father. Blue sex chromosomes are Y chromosomes, in pink sex chromosomes are X chromosomes. So in this case, this did come from the father, this did come from the mother. Alright, so the first thing that we're going to do is crossing over. We're going to have some random crossover events. You should note that this occurs in prophase 1 of meiosis. This happens right off the bat. People love to ask that question. So the way that crossing over is going to occur is chromosomes cross over and form a synapsis like that. So in theory, these two are going to swap parts, right? So how are we going to show that on socks? We have little fabric bands. So the pink chromosome is going to get just a little bit of paternal DNA, and this chromosome from the granddad is going to have a little bit of maternal DNA, right? And so this happens sort of randomly at different locations. So we're going to do the same thing with this chromosome. We're going to give this one. On a larger table, you can probably do this a bit more efficiently. We're going to do a little bit down here. Say that this chromatid crossed over this time. Crossing over is a bit complicated. Whenever you get to genetics, you'll see a bit more on that. But for now, we're going to keep it simple with little fabric bands. Very nice. And then we have one more. Let's put it here. We'll do it at the bottom. I'll make this crossing over site a little bit closer to the centromere, where the Velcro band is. Alright, So crossing over tends to be what kind of blows genetic variation out of the water, because you can mathematically calculate a lot of other stuff, as we'll see, but crossing over is pretty damn random. So this is what we got. Okay. So note how we didn't really cross over the sex chromosomes. They do have a little bit of crossing over, but it's minimal, so we're going to ignore that. So now notice how every single chromatid is different. None of them look exactly the same because of the bands that we have on them. So, yes, they're all different. So whenever we pull these apart, we're going to have them all different. There are cases where no crossing over, so say that we didn't do these two bands here. These would be what we call parental chromosomes. They're exactly the same as the parent. However, chromosomes like this, where crossing over did occur, we call recombinant chromosomes. So in this case, we made all recombinant chromosomes. So this is a very variable child to be. So notice how crossing over kept the structure of the chromosomes the same. They all still look like socks. And the location of the genes are all the same. However, the alleles that are in these crossed over regions might be different. So the sequence of alleles along the chromosome have changed. All right. So the next thing that we're going to show is random alignment. So this is random alignment at metaphase. At metaphase 1 of meiosis. So what we're going to do is we're going to use our chance probability. So if we roll an even number, the blue chromosome is going to be on the left. If we roll an odd number, the pink chromosome is going to be on the left. So let us begin with the largest of the autosomes. So 5, that's an odd number. So that means that the pink chromosome is going to be on the left. All right. Let's we have to sort of hang them off the table like that. All right. So with the shorter autosomes, we have an odd number. So pink is still going to be on the left. Notice how I could randomly align them how I want to, but we're trying to model how this is actually quite random. So you have no say in it. And then 6, so now it's even, so now blue is going to be on the left. So here we are at metaphase 1. The homologous pairs are all lined up, and they're going to separate as such. So that's random alignment. So if you were to calculate this with 3 different pairs, that's 2 to the n, so 2 to the 3 equals 8 different combinations. If you did this with humans who have 23 pairs of chromosomes, that's 2 to the power of 23, which comes out to be 8,388,608. So already, without crossing over and without random fertilization, your existence is already more than 1 in a million, just through random alignment. So, taking note of these, this father is going to give what will become gametes on this side, we'll give them a Y chromosome. So this will create sons, and then these will create daughters. Right? So remember that males receive their X chromosome from their mothers because the father has to give the Y chromosome. And remember that daughters get their X chromosome from their father. Right? Sometimes that can be a little bit confusing. And remember that the X chromosome is pink because it is the X chromosome. It's not the Y chromosome, which is blue. This is sort of a flaw in the model, so just take note of that. Alright, so what we're going to do is, this was metaphase 1. We're going to go through the whole works of meiosis 1, and then we get to meiosis 2. And so meiosis 2 is essentially very much the same as mitosis, except for that we have already become haploid with meiosis 1. So just like mitosis, the centromeres are actually going to pull apart. So chromatid disjunction occurs, and there we go. We have that. And then chromatid disjunction occurs here. And there we go. So now we have 4 gametes represented by these stalks. Alright, so in this case, we have a father, right? So we have 4 different sperm. 2 have Y chromosomes and 2 have X chromosomes. If this was a mother producing eggs, they would all have X chromosomes. Alright, so what's the same about this as the beginning? We still, or what's the same about all of these? All of them have 3 chromosomes, right? They're haploid. They only have half as much, with 2 autosomes and 1 sex chromosome each. What's different between all of them? Crossing over has made every single chromatid different, and every single one is unique. There's a bunch of different ways that you could word how they're different, but each one, none of them look exactly the same. So these are 4 very unique sperm cells. Okay, so for random fertilization, we're going to bring our dye back out. And in this case, if we let's say we're just going to have 1, 2, 3, 4, and we're going to roll to see which one gets picked. 5 or 6, we'll say that this parent didn't make it. The parent didn't make it. Just kidding. 2. This is the gamete that gets chosen. So we're going to go on, this sperm makes it to the egg, and boom, this father has a nice baby boy. Great. So whenever we would do this in class, everyone would be doing this simultaneously, and we would bring a sperm and an egg together, right? So in this case, we're just going to pretend that this other gamete came from a mother, since it has an X chromosome. We'll just pretend for a little bit. And so now we're going to fertilize this sperm that comes from the father, and this which we're now going to pretend came from the mother egg. So we bring them together, boom, 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 and we have a new zygote. A new child is born. Alright. So what's the same compared to our beginning cell, and what is different? Well, at the end, following mitosis and fertilization, we come back around to a diploid cell. So on this life cycle, we could show that everything beyond fertilization and before meiosis is diploid, and everything after meiosis is haploid, right? So we're diploid. We have 6 chromosomes once again. What is different? Well, we could have had a different sex of the offspring, but we didn't. We ended up with a male again, right? And we also now have recombinants. Before, we just had the parental chromosomes, but now all these chromosomes are beautiful and different. So this was meiosis. There are 3 things that make meiosis have variation: crossing over, random alignment, and random fertilization. And this is sort of the segue into the next semester's material, where genetic variation fuels evolution. So although this is a molecular process that we cover now, note that this is the gateway to a lot of the biodiversity that we see in the world around us. So just something to think about.